Hello students, in previous part we have discussed about the translation process. Now in this part we will discuss on gene regulation or regulation of gene. We have discussed about many processes such like as transcription, translation and different organisms such like as prokaryotic transcription, translation, both processes are occurred in the cytoplasm. But in eukaryote, transcription occurred in the nucleus, whereas translation process occurred in the cytoplasm. So during these processes, there are need to regulate the gene. Therefore, so that there is no any variation or mutation can take place. So the topic is gene regulation. There is need to regulate the genes during the different processes. As we know that there are two types of organism on the basis of nucleus membrane. The organism in which nucleus membrane present, those are called eukaryote, and in which nucleus membrane is not present, those are called prokaryote. In eukaryote, gene regulation process occurred by the multiple phases, multiple processes. In eukaryote cell, the regulation of gene is controlled by multiple processes. So there are many processes by which we regulate the gene, and those processes are, this is occurring eukaryote cell. First, during transcription. It means transcript primary, enzyme primary, hnRNA. To transcript the primary hnRNA. First of all, the regulation of gene takes place in eukaryotic cell. The first phase is transcription. When the DNA change into RNA or mRNA, that is called primary RNA, that is called hnRNA, heterogeneous nuclear RNA. In this phase, second one, during processing, as we have discussed in transcription, after capping and telling, introns are removed and exons are addition. This removal or addition process, there is regulation of gene can take place, and that is called processing. So here is splicing process. What is splicing? Removal of intron and addition of exon. Removal of introns and joining of exons. The expressed units in this phase are also required to regulate the gene. And then this hnRNA means after the transcription process occurred, this transcript RNA then transferred to the cytoplasm from nucleus. At that time also regulation of gene required. So next one is to transfer the mRNA from nucleus to the cytoplasm. This is the third phase. After transcription, mRNA then transferred from nucleus to cytoplasm. So here in this process also regulation of gene required. And next is as the mRNA reaches to the cytoplasm, translation process occurred. So there is also need of regulation of gene during translation process. So we can say that during translation process. So we can say that in eukaryotic organism there are multiple phases. At that times we require to regulate the gene, and those multiple phases are: first one is during transcription, means DNA change into mRNA, and after that splicing, means during processing, during splicing process, removal of intron, joining of exon, and then this mRNA transfer from nucleus to cytoplasm. So to transfer mRNA from nucleus to cytoplasm, there is also required to regulate the gene. And as the mRNA reaches to the cytoplasm, the translation process started. So in the translation process, there is also required to make the code, sufficient code or the same ditto copy of the code. Translation process we require to regulate the gene. These are the multiple phases occurred in the eukaryote. But in prokaryote, we have seen that prokaryote origin, all the cell organs with the cytoplasm embedded. So whole process of translation all will occur in the cytoplasm. In prokaryote, regulation of gene in prokaryote or prokaryotic cell. In prokaryotic cell, the whole process of regulation of gene is under control by the expression unit. Those are called accessory protein. Accessory protein, and these protein may consist many genes. These contains many types of genes such like as promoter gene, regulator gene, inhibitor gene, structural gene. So one strand of DNA consists of many types of genes, that is called polycystic. In prokaryotism, all the regulation, whole process in the regulation of gene controlled by the accessory protein, which contain operator gene, regulator gene or promoter gene, inhibitor gene, and structural gene. Which contain one strand of DNA. So we can see that in prokaryotic polycystonic genes are present, because one strand of DNA consists many types of genes. So that is polycystonic. How the gene controlation can take place? There are two types of system in prokaryotic which regulate the gene. That system is called operon. What is operon? The whole process of in the prokaryotic cell, regulation of gene controlled by these accessory protein that contain this type of genes. That system is called operon. And operon is of two types. One is inducible operon, and second one is suppressible operon. Inducible operon that remains in normal condition off. It remains in normal condition off, and we need to turn it on. It remains off in normal condition, but it turned to be on. In normal condition, this operon system will remain off mode, and we need to change it to on mode. For example, inducible operon is lag operon. Lag operon is a inducible operon. And second one is suppressible operon. Suppressible operon is normal condition remains on mode and it turn to be off mode. In normal condition or normally it remains on mode, but there is need to be turned off. In normal condition it remains on mode and there is need to change it to off mode. But inducible is opposite to the suppressible. Normally it remains off, turn to be on, and this one remains normally on, turn to be off. For example, tryptophan operon. This operon is always remains normally on mode and we need to change it to off mode. In next part we'll discuss on how the inducible operon or lag operon will be function.